This video is going to be the first of two considering animal basic patterns and development. So the first lecture is going to talk about basic grades of organization, or kind of the levels of organismal complexity. Then we'll cover the basic germ layers or basic tissues that and how that varies in different animal groups. And finally, we'll talk about some of the early developmental stages that are responsible for the production of these basic germ layers. So let's first start talking about different grades of organization that we see in animals, or basically how complex their bodies are. Before we actually get into animal diversity, we'll briefly talk about the protistans, and specifically a subgroup of them called the protozoans, some of which are actually the ancestral lineages that have led to the formation of the metazoans or animals. Some examples of protistans include euglena, volvox, and these represent the protoplasmic grade of organization. They're typically just one cell, and all of the complexity is just associated with organelles within their body. Volvox is a bit of an exception to that. It's basically an amalgamation of cells glued together, but without much kind of cellular differentiation, so there's no specialization or differentiation of different cells in the body. When we get into animals, we start to see that there is this aggregation of cells, and the simplest animals are simply kind of like Volvox. They're just kind of glued together. They show a bit more cellular differentiation, however, and this is represented by the sponges. So most people put sponges in this level. However, sponges, as we'll see in the next case, could be argued to be put in the cell tissue grade of organization. But here, the earliest animals we're just going to talk about potentially being just basically a mass of cells with some degree of specialization, but without true tissues. So we're going to be kind of looking at this figure to map the evolution of basic body plans in animals. So all animals start off as a fertilized zygote that's created by a sperm fertilizing an egg to get you a diploid zygote. And then they go through a period of what's called cleavage. And cleavage can be as simple as just producing this ball, hollow ball or fluid-filled ball called a blastula, without much differentiation of cells into specific tissues and really no organ systems or anything. And that pretty much describes our sponges. But the third level of organization is the kind of transition from masses of cells to forming true tissues. So here we have aggregates of similar cells that are forming patterns that are different from other types of tissues. Some, again, say that sponges are approaching this and having incipient tissues, but the cnidarians are a good example of an animal that's in the cell tissue grade of organization. So that's what's represented here. So we have, again, the blastula that can go through subsequent development and differentiate two different tissue types. So an animal with two basic tissue types, or germ layers, are called diploblastic animals. And you see that these are represented by things like the corals, the sea anemones, the jellyfish, so the cnidarians, and then a close related group called the ctenophores. The two cell types that they have are called endoderm and ectoderm. So those are the two cell types in a diploblastic animal. So let's break this word down. One of the things I want you to be paying attention to is prefixes and suffixes, because they will help you understand what the word means if you know a little bit of Greek and Latin. So di refers to two. The ploblastic refers to a tissue, so it means two tissues. Okay, and these are ectoderm and endoderm. Ecto means on the outside, so this will form the outside of the animal. This is the ancestral gut cavity, so this is the inside of the animal, the endoderm. So again, using these to help you, derm refers to a tissue. But using endo and ecto helps to tell you where that derm is. Most animals, however, fit into these latter two organizational categories. And I'm going to kind of combine them. It's the tissue organ grade or the organ system grade. Really the only difference here is between 4 and 5 is that 5 is talking about, so 4 you may have, say, a digestive tract, But in 5, you're talking about an organism with a digestive tract that has very specific organs that serve very specific functions in that digestive process. Something like in a frog that would have a stomach and intestines and a trachea, an esophagus, as opposed to something like a flatworm that has a digestive tract, but it's pretty much just this sac without a lot of specialization of different parts. But that's a relatively minor difference. What these all share in common is they do have organs or an organ system that is much more complex than what was seen in the diploblastic animals. So these animals would be fit down here, and at this point, you see, they at early stage of development, they look just like diploblastic animals, but they go through subsequent developmental phases in which it's not shown here, it'll show in the next figure. They have a third tissue. So these animals are called triploplastic animals. They have 3 tissue types. They still have endoderm and ectoderm, but now if you had something in between the endoderm and ectoderm, an appropriate term for that would be something that means in the middle. Meso means in the middle, so it's the middle tissue. And this is what most animals are. So again, we have ectoderm here in blue, endoderm in the yellow. This kind of reddish orange in between here, that is the mesoderm. And we're going to see that in subsequent development in triploblastic animals, there are a lot of different things that can happen with this. It can just fill the entire cavity of the blastocele. So here we're looking at the blastula, and it's cut in half so we can kind of look and see what this view looks like. So this cavity in here is called the blastocele. Sometimes the mesoderm fills it up. There is no body cavity. Other times it does produce a body cavity of one form or the other, and we'll cover this later. But you can see that there's a lot more complexity potentially with the formation of tissues when you have mesoderm added to endoderm and ectoderm. All right, well, to really understand what can happen during development when you get to this stage of having endoderm and ectoderm and mesoderm, we need to understand the processes that happen during development. So we're going to start off by talking about what happens from a fertilized zygote to different developmental stages and how changes in each developmental stage can help us understand animal evolution itself. So fertilization is the first part. It has to occur before development can occur, and that is the union of sperm and eggs to form a diploid zygote. Now, sperm are haploid, eggs are haploid. That means they have one copy of each chromosome type, so that when they come together, when a sperm fertilizes the egg, you produce the zygote that is diploid, meaning it has the 2N or the diploid number of chromosomes. It has 2 chromosomes of each type. Now, fertilization in some animals involves copulation, but in a lot of aquatic organisms, they simply shed their gametes into the water column, and this means that there has to be some pretty significant chemical communication between the eggs and the sperm so that they can find each other and fertilization can take place. And then that chemical communication doesn't stop there. Sperm then have to make sure that they are entering the right egg, or in truth, it's more the egg being the filter here, making sure that the sperm that have contacted the egg are appropriate for delivering the right DNA, because you don't want to have sperm from another species fertilizing the eggs of a different species. So there are various challenges that the sperm has to bypass to successfully fertilize an egg. The first of these layers is called the jelly layer, and it's the thick layer kind of in the light blue here in this figure. It also then has to go, if it makes it through the jelly layer, it has to go through this purple thin barrier called the vitelline envelope, and there's a lot of chemical communication that happens here. And then finally it has to pass through the plasma membrane before it can deliver its DNA material into the nucleus of the egg. So let's talk about what happens here. This jelly layer, and I should have mentioned that the jelly layer, the functions of this are in large part it's communication with the potential sperm, but it's also protective. So it provides some protection against bacterial or fungal infection, attack sometimes by small predators and even large predators, the jelly layer oftentimes has toxins or bad tasting chemicals embedded within it to deter predators from eating the eggs. Okay, so anyway, if the sperm do penetrate the jelly layer, then it has to contact the vitelline envelope. And there are egg recognition proteins that are seen in the sperm that have to bond with very specific protein receptors on the vitelline envelope. So there's that chemical communication that then triggers the next stage in the fertilization process. If the communication works, then the first sperm that makes that communication, it induces the formation of a fertilization cone, which draws in the head of the sperm, which contains the DNA, closer to contact with the plasma membrane. And at the same time that it's doing that, as soon as that sperm, the DNA payload enters the plasma membrane, it triggers the formation of this cortical granule release. Now, what's the purpose of that? Well, remember, we want to form a diploid zygote. That means you just want to combine the haploid component that's in the egg with one sperm's haploid component. You don't want a whole bunch of DNA from different sperm. So the first one that makes it through there triggers these cortical granules to be released, filling this area. You can kind of think of it as a biological cement that then makes this a thick wall that additional sperm can't penetrate. All right, then once the haploid sperm gets into the haploid egg, we form the diploid zygote, and development can proceed. And the first stage of development is what is called cleavage. This is when you take the single-celled zygote and you split it into additional cells. So you go from 1 to 2 to 4 to 8 to 16 to 32 to 64 until you get this fluid-filled ball of very small cells called the blastula. Now, during this time period, there really isn't a lot of growth. So the size of the fertilized egg, which again more properly is called the zygote doesn't grow that much to the blastula stage. It's simply taking that material and splitting it into smaller and smaller cells that form this fluid-filled ball. Now, one of the things that is seen in an early embryo is a greater or lesser degree of yolk. Yolk is the nutritive material that the early embryo needs to develop. It provides the energy for it to develop. And there's a wide range of yolk in the zygotes of different animals. Some animals have a lot of yolk, and it's clearly concentrated on one end of the zygote, and we call that the vegetal pole. Then the opposite end of that is usually where the embryo develops. We call that the animal pole. So notice that this isn't a typo. It's not vegetable pole, it's vegetal pole. So the amount of yolk, as I mentioned, varies across different taxa, different species and major groups of animals. In some animals, there's not a lot. It's what we call isolecithal. So a sea star, for example, is an isolecithal egg. You don't have a lot of it, and it's pretty much evenly distributed gets evenly distributed as we go through cleavage, so that all of these cells in the blastula have about the same amount of nutritive material. Mesolecithal, a frog, has a pretty distinct animal pole and vegetal pole, so that as we see there's division, we see many more very small cells in the animal pole, and you don't get always complete cleavage into cells of equal size in the vegetal pole. In a telolecithal egg, something like you would see in a chicken, there's just a whole lot of yolk. I mean, the zygote is composed in large part of yolk for the developing embryo, and the vegetal pole really is kind of almost the whole thing, and there's just a teeny tiny little patch or plate-like structure on the animal pole where the developing embryo is going to be produced. Now, there is a fourth type of zygote called a centrolecithal egg. These also, kind of like telolecithal, have a lot of yolk, but it's not concentrated on one pole or the other. It's actually concentrated in the center of the zygote. Now, the amount of yolk influences the patterns of cleavage that you see during development. If you don't have a lot of yolk to go through, you pretty much have clear cleavage going through all of the remaining cells at each stage, so that at the end you get a blastula that's pretty evenly distributed with cells, and the cells have about equal amount of yolk. That pattern is called holoblastic cleavage. So holoblastic cleavage is typically associated with isolecithal eggs. Now, we talk about isolecithal, mesolecithal, and telolecithal eggs as categories, but realize it's really a continuum. And so the closer you are to having a true isolecithal egg, the more likely you're to have complete holoblastic cleavage. If you get to the extreme of like having telolecithal eggs, it's really difficult for cleavage to make it through all of that yolk, and really there's no point of it anyway. So we tend to see what's called a meroblastic cleavage pattern in that case, where the cleavage really only occurs in the animal pole, and you have incomplete or absolutely no cellular division in the yolk itself. So that's going to be most typically associated with telolecithal eggs. Again, mesolecithal eggs, they can sometimes have holoblastic characteristics or meroblastic characteristics depending on where they fit in this continuum. But on the exam, I'll give you a question associated with one extreme or the other. So if I say, what do we have? What kind of cleavage do we have associated with isolecithal eggs? You can say holoblastic. If I say telolecithal eggs, then that would be meroblastic cleavage. At the end of cleavage, you get the blastula, and it's just that hollow ball of cells that's fluid-filled, and that fluid-filled interior is called the blastocele. The next phase is called gastrulation, and this is where you have an invagination in one part of that blastula to form this cavity, a second cavity, which is called the gastrocele or the archenteron. And this is the developmental gut. This is the gut that's developing in the embryo. Okay, so we still have a residual blastocele here, but now we have a gastrocele as well. Also, now we have an opening. This would be, so if this is cut in half, think about this turned, put back together half, and then turned toward you, there'd be a hole here, right? That hole is called the blastopore. And later we'll talk about what the blastopore becomes in different animals. In some animals, it becomes the mouth. In some animals, it becomes the anus. As I mentioned, some animals, their level of organization is simply to be diploblastic, two tissues. And they have some basic organs, but they're not as complex as we'll see in the triploid organisms. Again, this would be represented by something like a ctenophore and an cnidarian. This is where we're starting to see the formation of these germ layers, these basic tissues, the ectoderm in blue and the yellow representing endoderm. Sometimes the gastrocele doesn't get much more complex than that. It's a blind gut system. Okay, so you have a mouth that also serves as the anus. We'll refer to that as an incomplete digestive system. In other organisms, there's continued movement of these cells so that this leading edge contacts the other pole of the developing embryo to make this continuous gut tube. That we call a complete digestive tract. And again, which of these becomes the anus and which of these becomes the mouth is going to vary in different lineages of animals. But also, as I mentioned in that previous figure, some animals continue development of and specialization of a third tissue. These are the triploblastic organisms, and that third tissue is the mesoderm. Mesoderm, meaning middle, forms between the endoderm and the ectoderm. And how the mesoderm forms varies from lineage to lineage. Sometimes it forms as outpockets of the gut. Sometimes it forms from the lip of the blastopore, as we'll see. All right, so we've talked about a zygote, then going through cleavage, to give you our blastula, and then going through gastrulation to produce in some organisms just endoderm and ectoderm, but in others mesoderm. I don't want you to memorize this figure. The point of this figure is just to show you how important mesoderm is. Mesoderm interacts with the ectoderm and endoderm to produce some pretty substantial organs and contribute to organ systems. So much of the nervous system in animals is made up of ectoderm, but some of the support structures like notochord is also going to be associated with that. The circulatory system, the muscles, the urogenital system is largely mesodermal in origin. When we get to the endoderm, obviously this is primarily associated with the primitive gut. So organs associated with the gut, the digestive tract, the epithelium of the urogenital system. So there is, again, some combinations of endoderm and mesoderm working together in these systems. All right, so in review, we summarize kind of the grades of organization in animals. And the simplest animals, we'll talk about the earliest animals, the sponges, really are not much more than just an aggregation of cells without a lot of specialization, some cellular specialization, but not really organized into tissues. Then we'll talk about the tissue level of organization, in which you start to see this differentiation of tissues into endoderm and ectoderm. And in some of those, you can see some pretty simple organ systems being formed, but without specialized organs in that system. But that next level is where we really start to get more complex organs and organ systems, and this is typically associated more with the triploblastic animals. Again, this is a continuum. They're kind of breaking it up into categories, but just realize that we're going to see this evolution of complexity occurring in animals. And we'll talk about, the sponges first, then the diploblastic animals, but most animals fit into this triploblastic group with organs and organ systems. So the germ layers that are associated with these two layers are endoderm and ectoderm for diploblastic, and triploblastic animals, endoderm, ectoderm, with mesoderm in the middle. But again, if you're just the aggregation of cells or incipient tissues and sponges, you don't have any true germ layers or tissues. Then we started our discussion of development. We're going to continue this in the next lecture, but I just wanted to kind of show you some of the early stages of growth that comes from after the fertilization process, where we form the zygote. Fertilization itself is a chemical communication between sperm and egg, and then the sperm has to penetrate the egg's outer layers. First the jelly layer, then the vitelline membrane, or sometimes referred to as vitelline envelope, and then the plasma membrane before it can deliver its payload of DNA to form the diploid zygote. And to prevent multiple sperm from getting into an egg, the cortical granules fill that area between the vitelline membrane and the plasma membrane to prevent additional sperm from getting through. Cleavage then is initiated once fertilization takes place and ends with the production of a blastula. And that contains that interior cavity called the blastocele. We talked about the yolk and the amount of yolk and whether it forms a vegetal pole, an animal pole, and it can be an isolecithal, not a lot of yolk evenly spread, kind of middle, or lots of yolk concentrated in the vegetal end. And then centrolecithal is kind of a specialized group. It's actually pretty widespread, though. The arthropods have centrolecithal eggs. But again, there's cleavage. The pattern of cleavage varies depending on the amount of yolk and the concentration of yolk in different parts of the body, such that holoblastic cleavage is more likely associated with isolecithal, and meriblastic cleavage is more typically associated with telolecithal. After cleavage, when we form the blastula with the blastocele, then we have gastrulation. So that invagination of one end of the blastula to produce the gastrocele, which is the primitive gut, and the opening into that is the blastopore. So with that understanding of the basic germ layers and basic organization in animals, we're going to next time talk about how some animals have body cavities, internal body cavities, others don't, and we call these coelums. We'll talk about the two major lineages that show some pretty significant developmental differences in how we go from a single zygote through cleavage to gastrulation and the later development into specific individuals. And these are the deuterostomes and protostomes. Then we'll also talk about how animals vary in symmetry and how this greatly predicts the basic behavior and lifestyles of these different animals. And finally, we'll talk about just some of the basics of the genetics of development, of how we get these animals to look so different.